Okay, I am giving this another go from the beginning. I've had some problems. I've had some problems with this, but without audio. So I'm gonna just go really quick through this, and if you have any questions, we can talk about it tomorrow. So this is really just a preview of what you'll be doing the whole semester. So they're giving you some tips as far as the different technologies that are utilized in studying cells and tissues and the molecules within cells. Three different strands that form cell biology: cytology, study of cells; genetics, the study of DNA and inheritance; and biochemistry, the chemistry behind interactions that make all cells able to function. Okay, so there's some historical information in the first part of this. It's really interesting to read, and I recommend you do read it. And they go over the cell theory, which we've studied in general biology, and it should be familiar to you. If it's not, really give it a second or third read here so you understand it. All right, and so the cell theory really got going after microscopes were invented and after microscopy became a more successful technique. So there were a couple of limitations that that the early microscopists ran across. One is that things weren't so clear, and that had to do with the resolution. They were not able to see fine detail. And the other thing was that at this time in history, they were, instead of trying to do experiments and seeing differences and changes, they were really just describing nature, which was incredibly valuable, but limited. So as time went on, different improvements were made to microscopes, the first one being adding two lenses, the eyepiece as well as the lens that is down where the sample is. So when there were two lenses, they were able to really increase both the magnification and the resolution. This is really beautiful images of just the enormous diversity of cell types. Most of these are SEMs, some just regular light microscopy. And I just think it's a way to get hooked on science, really beautiful science. Okay, so modern cell biology has emerged by these different disciplines being interwoven. And cytological aspects of science are better understood now because we have discovered things in biochemistry, and biochemistry is a more robust field because of information from genetics, for example. So it's how these different different disciplines can cross-pollinate each other and provide more information than just on the original one. And this you can see kind of how things were separated up until the last century. Let's stop here and talk a little bit about cell measurements. It really helps to understand units. Membrane, 7 to 8 nanometers, ribosomes, 25 to 30 nanometers, DNA double helix, 2 nanometers in diameter. Bacteria cells, 1 to 2 micrometers or microns. These are called microns. Animal cells, 10 times that of a bacterial cell, for example. Okay, so that has some animations with this information. Now, the micrometer is 10 to the 6 meters. Now, you probably are more familiar with a centimeter. So a centimeter is 10 to the minus 2 meters. A millimeter, 10 to the minus 3. Micrometer, 10 to the minus 6. And the next one that you'll run across is, this is the nanometer. So a nanometer is 10 to the minus 9 meters. And an angstrom is 1/10 of a nanometer, and it's very small, about the size of an atom. And angstroms are used in really small, to measure small molecules rather than cell structures. All right, so you had some homework where you looked at different applications of microscopy, and you had to identify the type of microscopy that would have been utilized to look at that, or that you would have wanted to apply. Now, not all of these questions were necessarily just one answer, but the idea was for you to go back and look in the book and see the variety and understand what the different kinds of microscopies are and what applications they are. All right, so in doing microscopy, you may have looked at cells or some sort of, a dot of pond water and seen what was in there. But if you're looking at tissues, you have to be able to section them very thin because you cannot look through a microscope at a thick section and see anything, because the light comes from the light has to go through the sample. So a tool called the microtome was devised, and it depends on the particular application. This is a common microtome that is used for paraffin embedded samples. So these are samples that have been dehydrated and they're embedded, and sometimes it's a very useful way to do simple staining. If you want to use frozen sections, you end up sometimes not with the same quality of the cell. Sometimes it gets torn in the process, but it's really quick. So you can just take a block of cells or a block of tissue and freeze it on this chuck, and the chuck goes inside this instrument, and you roll this around. So I think I want my pointer. You roll this around, just like with this, you roll this around. So if you're using paraffin embedded sections, there's a very sharp blade on this end of it. And so as you roll this, it goes ka-chink, 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 and every roll, you're able to cut off one very, very thin section. And it gives you enough tissue to look at, but not so much that it blocks the light. This is a nice diagram to think about the relativity of different cell types, organelles, etc. Now, as I mentioned, there's a lot of different kinds of microscopy, and your book does a pretty good job of that. These are the different types listed and described. One application of microscopy is to look at living cells. Sometimes you want to see if they're moving or how they're organized or whatever. So phase contrast or differential interference contrast microscopy are often the type of microscopy you'd like to apply. Fluorescence is a particular kind of microscopy. It can be done two different ways. A common one is it's called immunofluorescence, and you use an antibody. Oftentimes you want to recognize something on the surface of a cell, and so you either purchase or make an antibody from an animal that has a reaction to an antigen. So you purify that antibody, and then you can use it, you incubate it with the tissue, and you wash it off, and then you apply a chromophore, and the chromophore sits on the antibody and it lights up. So you look under a microscope and you can see if that cell type has that particular protein you're interested in. For example, a receptor. If you're looking at a nerve cell, you want to know if it's got a particular receptor possibly. So that's immunofluorescence. Sometimes you can actually generate a fluorescing cell or fluorescing molecule in a cell as you're growing the cell. So that's another kind of fluorescent microscopy. And that one is with this GFP, you can incorporate that gene so that the cell is actually producing the protein. Confocal microscopy is used with a laser beam to illuminate a plane of a specimen, and this is useful because you can collect those laser images. It will do them serial like that, and then you can combine those images computationally and be able to look at your tissue and in a broader way. So these are different kinds of microscopy using light, just a comparison of what you see. And you can see with phase contrast and differential interference contrast, you see more contrast between the substratum, the plate here, and the cells. So compare these two with this one, for example. And there you can't see the cell outline and the inside of the cell as well. So these all three can be used for live cells. Now, this stained bright field here is not something that's compatible with living cells. The cells have to be fixed for that. But these you can look at some sort of living cell process. Now, development of the electron microscope was a huge step forward in understanding tissues and cells at a really fine level. And instead of light, the electron microscope uses a beam of electrons. And you're able to then analyze that data and understand the image. So not only did this increase resolution, it increased magnification by 100 times. So you can see how much, using electron microscope, so much more information was able to be extracted from looking at cells and organelles and tissues. There's two main types of electron microscopy. There's TEM and SEM, transmission and scanning. And scanning is done oftentimes to look at a whole organism or cell. And you can see unique three-dimensional information that way, whereas transmission, if you want to see inside of a cell, for example, look at the ribosomes, for example, you'd want to use TEM. So that's a fixation and a heavy metal staining. And scanning is a different method totally. You don't treat specimens with that strong stain. Now, there's a third kind that's used, and that is, it's called cryo-EM, so cryo-electron microscopy. And that microscopy, you can create an image computationally of something in 3 dimensions. Also, you can use cryo-EM to understand the structure at an atomic level of a molecule, of a macromolecule, a big structure like a ribosome or spliceosome or something like that. So in the past, we've only been able to establish structure at an atomic level using crystallography, which is limited. Some proteins just are not amenable to those conditions. So cryo-EM is another way to generate 3D structure of complex molecules. Here's a couple of TEMs and SEMs in the lower panels. Amazing structures, huh? Look again at the different sizes. So here they're looking at hundreds of nanometers. Here it's a whole cell. They're looking at 10 microns. So I posed a question here. So this is a question from your homework, and I think some of you thought about it and gave it your best shot. Some of you found answers in some source, and so I really hope that you're able to think them through and challenge yourself and get some wrong. You know, I mean, that's the only way to learn. Don't try to just get the right answer. Really try to understand it. So a scientist wishes to visualize the ribosomes of a cell. So that's a subcellular particle, very small. So you wouldn't use light microscopy. Would you use scanning electron microscopy, or would you use transmission electroscopy? Well, you want to look at the inside of the cell. So transmission electroscopy, microscopy would be the best tool there. A microbiologist wishes to examine the motility of a bacterium. So you want to look at something live. So remember earlier there were some slides that I talked about that, looking at the live cells, different light techniques for that. Immunologist wants to determine if a lymphocyte possesses a certain surface protein. Sounds like a great application for immunofluorescence, so fluorescent microscopy. D, virologist trying to determine three-dimensional shape of a virus. Try some SEM. And pathologist is trying to examine the cytoplasm of a cell for changes that result from viral infection. So that's probably another good application of transmission electron microscopy, wouldn't you say? Okay, so we've talked about cytology. Now let's take a look at biochemistry and how it fits into cell biology. All right, so biochemistry includes all of metabolism, and so all of the early findings of metabolism come under this umbrella of early biochemistry. They were able to show that yeast could make alcohol from sugar, and that just the inside of a yeast, the extracts of a yeast, could do the same thing. So that was kind of novel, that it wasn't a cell, it wasn't magical properties of the cell, it was really what was inside of it, and the contents of the inside of the cell. And they were able to discover that these things, these processes producing alcohol from sugar was a result of enzymes. And here's a little history on that, on the metabolism aspect of biochemistry. Now there's some biochemistry tools that are used, subcellular fractionation, and that's where parts of the cell, a cell is ruptured and spun down different speeds, different times, and you're able to see where different organelles will fractionate depending on the speed and things like that. And we'll talk about that a good bit later. We talked about that a good bit in biochemistry, so hopefully that's familiar to some of you. If you want to go another step and isolate really tiny things, like certain molecules, ultracentrifuges are available, and they can both preparatively and analytically fractionate your samples containing small molecules. Okay, some more methods in addition to centrifugation: chromatography, electrophoresis, mass spectrometry. Mass spectrometry is commonly used to characterize proteins. It does have some other applications, but that's all we're going to concentrate on in this class. Electrophoresis, we mentioned SDS-PAGE, polyacrylamide gel electrophoresis, using SDS as a detergent, and this uses an electrical field to separate proteins, DNA, or RNA through some sort of gel medium. There's different kinds of electrophoresis. There's the page that I mentioned, polyacrylamide gel electrophoresis, and that's used for proteins. Agarose is more commonly used for DNA and RNA. Now, chromatography it's a very flexible, adaptable method of separation. A lot of times it's analytical, sometimes it's preparative, meaning you can collect your samples and further analyze them. So there's lots of different ways to do chromatography. Here's an example of chromatography. This is probably just paper chromatography, separating different plant pigments. And this is the polyacrylamide gel electrophoresis, SDS polyacrylamide gel electrophoresis, with this is called the standard lane. So there's a comb at the top of this. Individual samples go in these kind of wells here, 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 and they're trying to compare these 6 samples, looking for differences. And this is typically an analytical technique. Let's look at a couple bands here. Let's look at the bands in lane 3 and the bands in lane 4 in this section right here. So those have a different pattern, so that would be something that would be noted and probably analyzed further. And then to know about the right size, the standard lane here allows you to estimate it. I don't know what this one is actually. This may be 200 and this be 116. So it could be this area is about 150 kilodaltons, and that's how you size proteins. All right, so here's posing a question. Looking at the different techniques we just talked about, you want to purify mitochondria. What's the best way to obtain this sample? Well, you're going to lysis cells so the mitochondria are free from membranes and all that. How do you want to get the mitochondria separate from the cytosol or the nuclei or whatever gemisch is in the lyse cells? Well, we talked about these 3 being pretty much analytical techniques. So you're not going to obtain a pure sample of mitochondria. And also, these lower 2, actually all 3 of these, are typically for smaller entities like macromolecules or molecules or peptides even. Whereas this, you want to isolate a whole organelle. So centrifugation would be the best method of doing that. All right, and the third strand is that of genetics. And early genetics involved Gregor Mendel's study with peas and the different other scientists that were involved in understanding heredity, essentially, and understanding or naming that the heredity factors are called genes. We know them as genes. So there were these factors that impacted heredity that we call genes, and there was also something that they could see through the microscope that later became known as chromosomes, and that the chromosomes were composed of the genes. So they had to really connect DNA to chromosomes to heritable factors, and then look at the components. What was DNA? What were the chromosomes made of? And they were able to establish that there were 4 different, something called nucleotides by the 30s, and then later identify proteins that were separate from this DNA material, and that proteins were composed of amino acids. So originally they thought there wasn't much difference between proteins and genes. And, you know, with time, they established the relationship between genes and DNA and chromosomes and protein. A lot of this involved Watson Crick and Rosalind Franklin through their work with crystallography of DNA. We talked a little bit in class about the concept of central dogma, DNA to RNA to protein, transcription, translation, and they've also included here, which is kind of cool, that DNA replication is a process that involves these three components. So that's where DNA has to be duplicated in order to divide and to make two cells, because each cell needs DNA. And you remember different kinds of RNA. I don't need to go over that. Exceptions to the central dogma include viruses with RNA genomes. So not all proteins, for example, are made from DNA to RNA to protein. Some organisms have RNA genomes, and so the RNA has to either be translated or converted to DNA and then go through central dogma. In vivo, the enzyme that does that, so in the organism, reverse transcriptase, and that has been utilized in vitro, so in lab settings, to be able to make RNA, to make DNA from RNA. So when reverse transcriptase was first identified, and it was manufactured in bulk, and so now you buy it, and it's just made a really huge difference in molecular biology science. Okay, I love this cartoon, study this cartoon, understand the different parts, and understand these descriptions of what happens with these arrows. See these arrows? Well, these are all processes. And you should be able to tell me about those processes. You should be able to tell me if you were to see this cartoon without the descriptions 1, 2, and 3, you should be able to offer me those descriptions. Eek. Sort of go back and forth. Okay. All right, 3 different terms I want you to know here. Recombinant DNA technology, you put new genes or new strands of DNA together through restriction enzymes and ligases. It doesn't say that. So first you take DNA, you cut it into smaller pieces, then you can rearrange it, and you got to stick it back together with ligase. And in that way, you can create recombinant DNA molecules. The second term I want you to be familiar with is DNA cloning. And that is just making a lot of copies of a particular sequence. If you wanted to express a particular gene and culture and make a bunch of protein or something, you would do some DNA cloning. DNA transformation is where you introduce DNA into a cell so that that cell can do what that DNA is coding for. All right, so different types of sequencing. There's kind of the normal sequencing where you sequence each base at, G, A, T, C, T, A, G, blah, 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 something like that. But there's also sequencing technology that's been developed through the Human Genome Project 20, 30 years ago, where the entire genome can be sequenced. So that's kind of a new, exciting science. In order to utilize sequencing information or a lot of computational information from even microscopy, you have to apply bioinformatics. And this is a computer science that you need to be able to make sense of all of this data. Genomics and proteomics are the 2 big omics sciences. There are other ones, and those certainly use tools that have been created and are publicly available through NCBI, high throughput methods where you have a lot of genes that you're sequencing at one time. Those methods are called high throughput. So you do different kinds of computational methods to analyze that data. This is a microarray, and microarrays are used to look at the expression of a gene. So you think about DNA being composed of genes. You think about transcription making RNA, and then the RNA is translated into DNA. But say you have a new drug and you want to know if that new drug is going to cause changes in the genes that it's expressing. For example, if you're trying to treat cancer, you want to know if a new drug might suppress the proteins that are involved in DNA duplication, replication. So how this data is used, it is known what all of these genes are. And this information tells you that this gene, and you have a map that represents this matrix, this gene, more of it is made in the presence of this drug than without the drug. This slightly orange, and there are some brighter red ones like this, indicate that there is less of that gene being expressed, less of the protein, less of the gene. Whoops. And the yellow says that there's no difference. So this is a really handy tool to look at gene expression. So microarrays look at gene expression. More omics, transcriptomics, metabolomics, lipidomics, ionomics, and we'll see where that goes. But all of those generate a tremendous amount of data, and it's the bioinformatic tools that are useful to understanding that. The next part has to do with the nature of science. And those of you that are doing the senior seminar might want to read this. And so just gives you a little different perspective on what science is and isn't. Let me go back to this one, looking at what facts are, looking at what a hypothesis is, the need to use peer-reviewed sources in understanding journals. All right, and the last part, I wanted to look at model organisms. And all of you will be selecting or assigned a model organism, and then you're going to tell the class about it. So I'm looking forward to that because they're a lot of fun. You learn a lot about all the different kinds of science that have been done using model organisms. Tissue culture is common, usually, well, not necessarily usually, but oftentimes, especially with human medical science, instead of going to an animal, you're going to want to start out with cell culture, and so you understand the process a little bit better. And then you might go to a whole organism. And there are certain things a model organism has to do to make it a good model organism. It has to be widely studied, well characterized, and easy to manipulate. Different model organisms have different advantages for study. And so we don't have an unlimited number of model organisms. We have about 10 that are common. These are 6. You've got bacteria, E. coli, yeast, fruit flies, roundworms, mice, and this is a mustard plant, Arabidopsis. I don't know if I said that right. So each one offers something unique as far as understanding a cellular process or a biological process better. Okay, so there's this question, and I want you to understand the difference between a meter, a millimeter, a nanometer, and an angstrom. So I'm going to let you think about this, and we'll talk about these questions in class tomorrow. Okay, and I gave you some information earlier about measurement. So I think we're caught up with this one, and thank you very much, and I'll see you tomorrow.